Hi guys, good morning. My name is Carl Artalia. Today we talking about phylum ctenophora. Phylum ctenophora is the animal phylum whose members are radially symmetrically diploblastic or possibly tripoblastic, usually ellipsoid or spherical in shape, possesses by coloblasts and have myriadly arranged combros. Class names are not listed here as a higher level taxonomy is currently under revision because it is traditionally class designations do not reflect monophyletic groups like ctenophor like ctenophores are major predators upon larval fish and zooplankton. They may prey on small crustaceans, also known as phylum arthropoda, and the larva stage of the various fish and jellyfish species and oysters. Some are carnivorous on other ctenophorous or gelatinous animals in other groups. There is an evolutionary relation between ctenophora and cnidaria. Nearly all ctenophores are bioluminescent. They give off excess energy in the form of light instead of heat. The functional significance of bioluminescence includes mate locomotion and species recognition, lurking of prey, and the startling of would-be predators. Adhesive structures called coloblasts, 8 rows of ciliary bands called comb rows for locomotion. Morphology. The morphology are most ctenophores are round or spherical in the body shape. They generally have 2 large tentacles and 8 rows of comb plates. Their tentacles have the adhesive coloblasts to capture prey, and they have a stomach, mouth, parts, and canal pores. They have a nerve network that is rather complex and an optical sense organ. All ctenophores are hermaphrodites, and most reproduce sexual. They generally range in size from 0.6 centimeters to 30.5 centimeters, like this from the picture. Did you see they have apical sense organ, tentacular sheaths, parts, and complete, and have a mouth? The ctenophora structure, similar in structure of Cnidaria medusa, poorly studied due to fragile nature. Ctenophore structure has a body surface has 8 rows of comb-like paddles. Comb paddle has thousands of cilia used to swim. Apical sense organ. Apical sense organ are located on the arboreal surface, control beating of cilia, statolith, and balancer. So that means they are balanced while the ctenophore have doing some swimming mechanism like dome, tooth, balancer, and column rows. Coloblast. Coloblast has a sticky retractile tentacle used to catch prey. Coloblast has a discharge and an adhesive thread which is sticky to the watch. Some SPP lack tentacles and the body is studded with coloblasts. Look at the picture. There have been head, granule, spiral filament, and straight filament. Coloblasts. The morphology of coloblasts. Coloblasts are microscopic sticky structure used to adhere to the prey of ctenophora. Usually the class of Syndipida, and Lobata, and have coloblasts on their tentacles. After a prey is caught by a coloblast, the tentacle is contracted and the prey brought by the predator's mouth. There the prey is released and ingested. The digestion. Feed on plankton, other ctenophores, and other cynidrian gels. Gut extent through the entire body. Mouth is at the oral end. Anaphore is at the aboral end. In addition, the gut extends through the entire body. Mouth is at the end of the oral end, while the anal pore is the aboral end, under through the stomach. Reproduction. The reproduction of ctenophora can rapidly regenerate lost or damaged parts. Usually, hermaphrodites develops developmental stage. Cdipid larva develops into an adult. Compared to colenterate, the difference between of the colenterate: 1. Only 2 tentacles, 1 statocyte, no nematocyte. Coloblastiki adhesive cells on tentacles for feeding, no polymorphisms, never colonial, has an anal open. The similarities: never net, same 2 body layers, epidermis and gastrodermis, masoglia, also known as have a muscle. The embryology of ctenophoras. When the egg is fertilized, development of the embryo begins. Cleavage occurs completely but inequally, first reproducing the 2 cells, then 4, then 8, and so until the embryo is full development. The embryo forms within the egg cell. It develops double rows of cilia, a pair of lateral tentacles, and a large optical sense of organ. The intradermal of the gastrovascular system undergoes epiosexis. Epiosexis involves the flattening and extensions of the ectoderm germ layer in the gastrovascular system. Soon the cilia begin to function and the developed larva breaks in the eggshell and enters the water. In the following page are detailed steps to the process of initial fish development. The development of embryology. First, there was cleavage to look at the picture. There are cleavage to cleavage 3, then go to the cleavage 4, which is the formation, and the cleavage 2, which is the 2-cell formation, and the cleavage is into the 4-cell formation. And before the cleavage there's a 8-cell formation, and after that there was a 16-cell formation, and after there was a collide between 16-cell formation. After that, there's been 32-cell formation into the stereoblastola to gastrulation. After the gastrulation, there has been a gastrulation 2 formation with each steady, and then gastrulation has to transfer it, and after that is gastrulation 4 was completely into a cipidly larva formation. Look at the look at the picture. They have been formed into a cipid larva. Thus, ctenophora diversity. The ctenophora diversity, most species live in the open ocean and are not well studied. New studies use submersibles and divers to collect specimen, eliminating specimen destruct by the fast towed. Next, phylum ctenophora. There have been 2, order Labota and order Beroida. Order Labota is possessed long tentacles to the life cell retractile in a nature. Labota morphology have a pair of muscular lobes extended from mouth tentacles and grooves on lobes. Have auricles between lobes and mouth auricles and have cilia to create current and flash prey into the mouth. Two comb rows on each lobe and two on each side. Between the lobe combs movement depend on nerves rather than the water movement. Some can clap combs to push water rapidly and move opposite, generally prey on plankton. The order Buriod. The order Buriod has a lack tentacles or oral lobes. Can consume prey substantially larger than itself. Cylindrically flattened body and has no tentacles. The Barod's morphology. The Barod's morphology order, also known as Noda, it has no feeding appendage, but parmex have a macrocilia. Macrocilia are large bundles of cilia that work to bite off pieces of whenever is being consumed. Prey on either ctenophoras when not eating. A reed shuts the mouth by connecting with the other part of the reed. That's all. Thank you.